We know from previous research done in the myeloma field that monoclonal gammopathy of unknown significance and smoldering myeloma, which are the precursors stages to multiple myeloma, are influenced by the changes in, for example, the immune cell populations and also other markers that also result in reduced tumor immunosurveillance. So we already know that reduced tumor immunosurveillance can lead to this progression to the later stages of myeloma. And because of that, we actually designed a study to characterize these changes and describe the changes in the evolution. And I think that we have to discuss first that the way we did our analysis is we analyzed the first 100 patients in our prospective observational study. And 20% of the patients that had a smoldering myeloma in the study progressed to multiple myeloma, most of them, and then 2 of them progressed to AL amyloidosis. And when we looked at all these changes, what determines how patients will progress is we observed first an interesting observation that somatic mutations, so certain somatic mutations, could increase the risk of progression. And that is something that has been described before in smoldering myeloma. And so this is a confirmation of that. And we did, for example, whole exome sequencing in 60 samples from 65 patients. And the patients that did progress to myeloma were the ones that had most of the somatic mutations. And so I think that this was an interesting finding. We also analyzed in 8 of these patients that had a smoldering myeloma at baseline and then at progression to multiple myeloma in markers, immune markers of interest. And we saw that, for example, PD-L1 and PD-L2 and HLA-G were increased when compared to baseline. And these are just markers that reduce immunosurveillance. So they reduce the power that the immune system has to detect these myeloma cells as foreign and attack them. And so this just goes along with some of the observations that we've had. I think that some of the new observations that we have seen in this study that may have not been described before are, for example, an association with something called an interferon gamma signature with progression. So the patients that progress to multiple myeloma have a regulation of certain genes that are associated with reduced immunosurveillance again. And these are, for example, TIGIT and LAG3. And I think that these are important because there could be potential therapeutic implications for these patients. There's a lot of difference in the immune cell composition. So the composition of all the different types of B cells, T cells, macrophages, dendritic cells changes when patients are in the smoldering stage or MGUS stage and have not progressed versus those patients that progress at baseline. And these changes include, for example, a lower number of NK cells or activated dendritic cells, which are associated with a more active immune system. And for example, in the patients that progress, at baseline, they have higher resting dendritic cells, which may be less active. They have more macrophages. So all of these things are good to know because, again, you can look at these changes and you could potentially think about therapeutic interventions to modulate these changes to make, for example, a type of cell more prominent than others if you think that those are important in the progression. And so I think that these are very important findings. And I think those are the most important findings that we found in our study. And it's really just a growing body of evidence that the somatic mutations, gene expression, the immune cell composition, and certain immune cell markers are important in the progression to multiple myeloma. And I think that all this is very new over the past few years, and it may lead to new therapeutic agents in this field.